Hello students, once again welcome back to our biology class. Myself Dr. Raghavendra here. In the earlier class we were discussing about the classification of enzymes. So enzymes are basically six variety of or six categories as such, six classes of enzymes. They are mainly oxidoreductases, then they may be the what we can say that transferases, then they are hydrolases, they are liases, then isomerases as well as ligases we have discussed. So a small concept is still remaining in this chapter, that is all about cofactors. Let us understand what is a cofactor and what is the use of these cofactors. So when talking about cofactors, so cofactors as such, the basic structure of the enzyme reveals that this is the enzyme. So enzyme and its protein totally yes. The protein part we call it as the apoenzyme. Apoenzyme. So it is a protein. So they do have what we can say that non-protein part. So we can call them as a non-protein part, and they are called as prosthetic groups. Prosthetic group, or it is a cofactor. Okay, so the prosthetic group is a non-protein part, non-protein. Okay, so the protein and non-protein part and the presence of the cofactor, the importance of cofactor apoenzyme. So thus the prosthetic group as well as apoenzyme together, it is called as holoenzyme. Holoenzyme. So the role of cofactor actually attached with enzyme, so to the surface other than active site. Okay, so active site is mainly meant for, also called a substrate binding site. Yes. So presence of this, mainly this is what we say the active site or substrate binding site. Fine. So prosthetic group, or we can call it as cofactor. What is the role of cofactor? Cofactor mainly increases the efficiency of the enzyme. So cofactors increase the catalytic ability of the enzyme. Cofactors increases catalytic activity or efficiency of the enzyme. Efficiency of the enzyme. So the presence of cofactor, yes, play important role that gives the ability or efficiency or strength to the enzyme to accelerate the rate of or to increase the, we can say that, conversion of substrate to the product. So the catalytic activity is enhanced or increased by the presence of these cofactors. So cofactors are basically three types. Let me mention here. Cofactors can be of three types. One is we call it as a prosthetic group. Prosthetic groups. Second one is mainly they are the coenzymes. The third is metal ions. So When I talk about prosthetic group, yes, it is a non-proteinaceous, non-proteinaceous, but the coenzyme can also be non-proteinaceous, and yes, these are mainly prosthetic group as well as the coenzyme together. Yes, they can be the organic substances, whereas metal ions definitely inorganic substances. Well, so one simple example for the prosthetic group. Yes, let me give you simple example. So prosthetic group. The hydrogen peroxide H2O2, yes, can be converted into H2O plus monooxygen. So this biochemical reaction is mainly catalyzed by peroxidase, and this peroxidase enzyme. So it has prosthetic group heme. Okay, heme is the prosthetic group with this what you can say, hydrogen peroxidase. Then yes, coenzymes, they are most important and essential. Yes, biomolecules only we can say that. Example includes NAD, nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide, One more is there, NADP, nicotinamide adenine dinucleotide phosphate, nicotinamide adenine dinucleotide phosphate. Other than this, vitamin niacin also act as one of the most important, what you can say, the coenzyme. So when you look at metal ions, yes, definitely this is the enzyme structure. Here what we find a prosthetic group, and here generally the substrate bind over here. Okay. So other than the substrate site, there is some more part on the enzyme where these prosthetic groups or cofactors are attached. Now metal ions as mainly we can say that acting as a cofactor. Let me tell you a few examples. Metal ions as a cofactor. One simple example, especially the Zn2 plus act as cofactor for the enzymes, proteolytic enzymes such as proteolytic enzyme carboxypeptidase, and then one more enzyme such as carbonic anhydrase. In these enzymes, zinc is mainly the metal ion which is acting as a cofactor. Yes, there are several other kind of metal ions. Let me mention those also, which act as a cofactor. What are those? Let me mention here the other, such as mainly here you are observing such as Zn, Then Fe2 plus, yes, the iron, yes, mainly act as a cofactor for the enzyme catalase, and then Mn2 plus indole acetic acid oxidase. For this enzyme, it acts as a cofactor. And then there is one more, what we call it as mainly, yes, MO2 plus, molybdenum 2 plus, that act as mainly nitrate oxidase. Okay, so nitrate reductase. So in these enzymes, mainly Fe2 plus acting as a catalase, Mn2 plus acting as a mainly what we can say that for the indole acetic acid oxidase, and then so mainly molybdenum 2 plus mainly metals are acting as a cofactor for nitrate reductase. So thus the metal ions are acting as a cofactors, and then they mainly help in what we can say catalyzing these reactions. So here one more important metal ion. So Ni2 plus. So Ni2 plus. This act as the cofactor for the enzyme urease. So thus we can say that mainly the cofactors are those important kind of what you can say the substances attached with the protein part of the enzyme. So protein part of the enzyme, what it is called as the protein part of the enzyme is called the apoenzyme. To the apoenzyme, what is attached? It may be coenzyme or it may be a prosthetic group, that we call them as the cofactors. So cofactor plus apoenzyme together, it's called the holoenzyme. Then only the enzyme will be more active and its catalytic activity or efficiency increases. So thus here hydrogen peroxide is there that forms in the every cell. So it is further converted into water and monooxygen by the enzyme peroxidase. For the peroxidase, the prosthetic group is heme, and then mainly NAD, nicotine amide adenine dinucleotide, nicotine amide adenine dinucleotide phosphate, NAD, NADP are abundantly seen in a number of the steps such as in the electron transport chain system. Vitamins such as niacin, and yes, here it is also acting as a mainly the coenzyme. Metal ions are acting as a cofactor such as a zinc act as a cofactor for the enzyme, mainly the proteolytic enzymes we can say carboxypeptidase as well as carbonic anhydrase. And then we can say that the iron is acting as a cofactor for catalase, then Mn2 plus acting as indole acetic acid oxidase, and then molybdenum acting as a cofactor for the nitrate reductase, then nickel is acting as a cofactor for urease. Thus with these, yes, we understand the cofactors are very very important and essential, that gives the efficiency to the enzymes. Now we need to understand mainly enzymes and their applications. So few important points I would like to discuss before we end up this chapter: enzymes and their applications. So enzymes have lots of applications in the industry as well as in the medical healthcare. Enzymes have lots of applications in the plant sciences as well as animal sciences. So let us go ahead one by one the importance of enzymes. Well, so when we are talking about enzymes, yes, they are the biocatalyst, accelerate the rate of reaction. So applications of enzymes. Applications of enzymes. So the first one is mainly amylase. Amylase, one of the important enzymes that is known to convert starch into glucose. But this amylase also can be used for desizing of fabric. Okay, Other than that, yes, amylase also used in baking as well as in fermentation. Fermentation. Yes. Second, so protease. Protease is useful in meat tenderization. Meat tenderization. Other than that, yes, protease also used in, yes, deherring and baiting of hides. Yes. Then we can say that, yes. Then mainly decolorization and softening of animal leather. Okay. Number 3, pectinase. So useful in mainly, yes, softening of plant cells, clearing of fruit juice, fruit pulp, we can say pectinase. Yes, fermentation of coffee beans. Coffee beans. And then number 4, lipase. So these lipase are useful in mainly, yes, used in detergents to clean oil dirts in fabrics. Okay, well, number 5, lactase, curdling of the milk. Then number 6, streptokinase, the most important enzyme, yes, used as a clot buster. To clear blood clots. Inside the body. Well, so these are few essential and important, what you can say, applications of enzymes. So let me tell you, amylase is one of the important enzyme. Yes, we can call it as salivary amylase. What we have in our saliva that is useful mainly to digest the starch converted into the glucose. So this kind of amylase also can be used in desizing of the fabrics, softening of the fabrics. So cutting of the fibers, we can say desizing of the fabrics, baking, okay, so that spongy texture in the bread or the cake can come out, even useful in the fermentation. And then proteases, mainly the proteases act on the protein. So generally the beef meat is very hard, unable to chew, unable to digest. So what they do, overnight they treat that beef mainly with the protease enzyme. The meat becomes soft, easy to cook and eat. So meat tenderization, deherring, yes, once the animal is butchered or killed, and then the leather is taken, and that leather is full of hairs and hair follicles. So what they do, they soak that leather into the water, and also they add the enzyme protease. So the hair follicles easily can be get removed, and the leather, baiting of the leather or the hides, we can say softening and decoloration of the hides or the animal leather become very easy when they use the protease in an industrial scale. Pectinase, yes. So pectin is one of the important component of the plant cell wall. The plant cell wall is made up of what mainly? Cellulose, hemicellulose, yes. Even in when we crush or the smash the fruits, it may be the orange. So we get the pulp. So even we filter that pulp, we won't get the clear fluid. But when it is treated with the pectinase, that dissolves the pectin everything very clearly, and we get the, yes, very clear the fruit juice. We call as mainly the clearing of the fruit pulp using the pectinase. Fermentation of the coffee beans also by using the pectinase, so that further the coffee beans become very soft enough, and then its extract can be very much with a very good flavor. And then lipase, yes, the lipase is an enzyme that mainly break up the lipids. So used in the detergents, so that the cloth which are heavily dirt with the oil dirts, yes, that can be easily removed from the fabrics. So they use the lipase in the detergents. And then lactase. Lactase is mainly the milk further converted into the curd. So in the industrial scale, yes, lactase can be used. Streptokinase is most important. People undergo heart surgery, but there may be a chance of a formation of blood clots in the major blood vessels. So such blood clots can be very easily dissolved by using the streptokinase, what we call as the clot buster. So these are the major applications of the enzymes. So thus we are done with lots of important concepts related with almost every biomolecule. So thus we are ending the chapter here, yes, with the part with the application study of the enzymes. Well, so in the next class we will begin with some more new chapter. Until then, take care, study well. Thank you very much.